Many thanks, Rafi, and good morning to all of you, and many thanks for, now I had to hit the button for recording, or now it works. Thanks a lot for joining us today, and very welcome to Fernando Maestre, joining in today from Alicante. Very happy to have you here, Fernando. And just to make a quick introduction about Fernando, he has been receiving his master's and PhD from University of Alicante, and later on did a postdoc at Duke, returning to Madrid, the University of Rey Juan Carlos, where he still is a full professor, but currently on leave, and now he's back in Alicante. He has, I think these are really outdated numbers, but over 230 papers, which I found on the website, probably more in the meantime. He has gotten 2 years SNI grants, big one, starting grant, and a consolidated grant. I had learned yesterday that was the only person, or you are the only person in Spain holding 2 ERC grants at lifetime, I guess. And also last year, Fernando received now I have to go peek again about the name of the award, the Rey Jaume the first reward for environmental protection, which you may tell us more about what it really was about. I couldn't quite find all of it out. But yeah, I'm really glad to have you here as an outstanding ecologist. I'm very sure you have us a lot to tell on the global biogeography and ecology of soil microbes in a warmer and more arid planet. And with that, please, the floor is yours.
Okay, many thanks, Anita, for this very nice presentation and for the invitation. It's my pleasure to be here to talk a little bit about the research, some of the research that we do in my lab here at the University of Alicante, and in particular about the topic of the title of the talk, right? About the biogeography and ecology of soil microbial communities at the global scale, and the implications of this for climate change. You know, how this we study we don't focus only on microorganisms, but over the last decade in particular, we have done a lot of work with soil microbes at the global scale, and we are trying to understand some basic aspects of their ecology and biogeography, particularly in dryland ecosystems, which are the focus of research, and the implications of climate change, and particularly of increases in temperature and aridity conditions for these organisms. As Anita said, I work here in Alicante in the southeast of Spain, and I'm the PI of the dryland ecology and global change lab here at the University of Alicante. So I would like to start just by asking this question, which for you, I think there is no need for an answer, right? Because we all know how important soil microorganisms are, right? But despite that, we know their importance, and despite these organisms are the most diverse, particularly soil microorganisms, for the most dominate soil biodiversity. We don't know many basic aspects of their ecology and the distribution, particularly across large spatial scales. It's really surprising to see how little we know about many aspects of their basic ecology and distribution across environmental gradients, particularly when compared to other taxa like vascular plants or animals. And doing this, knowing more about this ecology and biogeography of these organisms is not only important because of their diversity and their importance within ecosystems, but also because they provide multiple and essential ecosystem service for us. Now, if we look at all the services that the soils provide to us, which are summarized somehow in this figure, microorganisms are involved in virtually all of them. And if we really want to know more or want to manage our soils better, we really need to know more about what microorganisms are doing. And as I say, we have been doing over the last decade quite a bit of work about the ecology and distribution of soil microorganisms at large spatial scale, and particularly in drylands. And I would like to start my talk by presenting some of the work that we have been doing, trying to study the environmental controls on the abundance and diversity and functioning of soil microorganisms across global drylands. And for presenting this work, before presenting this work, I would like just to very briefly introduce why working on drylands. And for multiple reasons. For me, one is clear: is I was born here in Alicante, in the southeast of Spain, and I was raised as a person and as a scientist in this type of dryland climate, in a dryland climate. So I have a personal and natural appreciation for this type of environments. But really, drylands are very important at the global scale. Collectively, dryland areas, which are those with an aridity index, which is one of the ways that we use to define drylands, this index is just the ratio between precipitation and potential evapotranspiration. So every region on the planet with a value of the aridity index below 0.65 is classified as a dryland. So collectively, all these places on Earth occupy more than 40% of the terrestrial surface and host over 40% of the global population. But to me, as an ecologist, it was really striking to see when I was studying biology and when I studied ecology and started to interested in this field, how all the examples I studied in my textbooks, all the papers that I started to read, they always came from non-dryland environments, despite these drylands occupy a large portions of our planet. So this is one of the reasons why me as a personally and in my lab we focus on the study of drylands with the aim to contribute to filling this knowledge gap that we have about the ecology, the overall ecology and the ecology of microorganisms in particular in this type of environments. And doing so, drylands are not only important nowadays, but they may become more important in the future. There is a lot of discussion going on right now about whether drylands are going to expand or not with increases in climate change. But because ongoing global warming, no matter what happens with rainfall, this is increasing evapotranspiration and this is increasing aridity, which is one of the, as I say, one of the main characteristics we use to define drylands. So one of the main questions we are addressing in my lab is try to understand how ecosystem properties, how organisms, how other structural attributes of dryland ecosystems and their functioning change across aridity gradients, because this can give us an idea of how climate change may impact dryland ecosystems. As we can see here in this map, I'm sorry, the legend is a little bit small. Many dryland areas are going to increase the degree of aridity because of ongoing climate change. So by understanding better how things change across aridity gradients, we may gain a better understanding about how these ecosystems may change due to climate change. And present some of the work that we have done on this topic. I would like to introduce the EPS Biocom Survey. This is the first global survey focused on drylands, and this was done thanks to my starting grant, the ERC grant Biocom, and other projects from many different colleagues all over the world. And this is a survey that I had the pleasure to coordinate, and that involved a collaboration with lots of colleagues from working in dryland ecosystems from all over the world. We were able to survey using a standardized protocol more than 230 dryland ecosystems from all continents except Antarctica. We got field data and samples from 21 countries. And as I say, we use standardized vegetation and soil surveys protocols, and all we gather on-site information on vegetation, on the diversity, the cover, the spatial pattern. We collected soil samples. All the soil samples were sent back to Spain, and we analyzed them in the lab for multiple variables, not only physicochemical variables to characterize soil environment, but also to multiple variables that act as proxies of carbon, nitrogen, and phosphorus cycling, which are good surrogates of multiple ecosystem functions and also of supporting ecosystem services. And we were able to get frozen DNA from over 80 of these sites. It was not possible to get to freeze samples quickly in many of the sites due to logistic reasons. You know, many of these sites are located either in very remote areas or in areas where there is no easy access to a refrigerator. Working in the middle of Africa, for instance, is not the same as working here in Europe. But still, we were able to get frozen DNA samples from over 80 sites that also provide a good representation of this database, this survey. I like to say that this survey is not perfect for anything, but it's good enough for almost everything. And it's because at the end we got a very nice characterization of vegetation, the soil properties, also abiotic factors. And for instance, by using remote sensing tools, we obtain additional properties and functions, for instance, like vegetation productivity. This survey provides a really nice snapshot of how drylands look like today, because our survey covered all major vegetation types. We cover a very wide range of environmental conditions, from sites with more than 1,000 millimeters of rain in Venezuela to sites with less than 100 millimeters of rain, for instance, in the south of Tunisia. We have sites very close to the sea in the southeast of Spain, and sites at more than 4,000 meter height, for instance, close to a volcano like this place in Peru. We cover with a survey also dozens of different soil types and conditions, environmental conditions, and this is a really nice survey that provides a nice characterization of how dryland ecosystems look like. So what we have done a lot of things with this survey. We have been publishing over the last decade also quite a few studies on this data, and I will just present some of the work we have been doing with soil microorganisms. The first paper we published with this data was a very simple one. We just look at the abundance and diversity of bacteria, soil bacteria and fungi, quantified using DNA approaches. MISIC, Illumina MISIC for the diversity, qPCR for the abundance, and in these panels, I just present a very simple relationship between aridity, which increases from left to right. Here we use 1 minus the aridity index to make it more intuitive. So aridity increases as the value of this method increases too. And in the left panels, we can see data for bacteria, and in the right panels, data for fungi. Either for diversity in the upper panels or for abundance in the lower panels, we got a linear decrease in the diversity and abundance of these microorganisms as aridity increases. It was a quite interesting result. We explored deeper this result, and we conducted additional structural equation modeling, which I guess most of you, or at least some of you, will be familiar with this statistical approach, which, as you know, with these structural equation models, we draw the relationships between the variables we want to model, in our case, the diversity and abundance of bacteria and fungi, and in this case, different predictors that also are not fully independent among them. So in this case, we include, of course, aridity and its relationship with important variables for soil microorganisms like pH, organic carbon, and vegetation cover, and also some climatic features and spatial coordinates, which we also include in our models. In some way, we include the information about the spatial structure of the data to control for spatial autocorrelation, because in a survey like this, sites within Spain, for instance, are not as independent among them that the sites between Spain and Argentina, for instance. And what was really interesting from this result is that we didn't find any significant link between aridity and the diversity and abundance of soil microorganisms. So does it mean that aridity is not important? Not really. But what we found in this model is that aridity has mainly an indirect effect. So aridity, increasing in aridity, reduce both vegetation cover and the amount of organic carbon in the soil. And organic carbon was the main predictor, which was strongly positively related to the diversity and abundance of both bacteria and fungi. This is not a surprise. We are dealing with heterotrophic organisms And of course, they need food, and the food comes from the amount of organic carbon in the soil. As you increase aridity, you reduce the amount of plant cover, and this reduces organic carbon inputs into the soil. So we can see here in these lower panels, which is a representation of the sum of the effects of the different predators, which is a measure of the importance of the predators, and we can see the importance of organic carbon, both for bacteria and for fungi. Climate is also important, of course. We're in drylands, and at the end, water drives biological activity in the system, but still, we can clearly see the importance of organic carbon for as a predictor to explain changes in the diversity and abundance of soil microorganisms across global drylands. We, as a follow-up of this work, we also explore the role of microbial diversity as a driver of ecosystem functioning and multifunctionality. This is a word that we are now using a lot in ecology. I guess most of you, if not all, are familiar with. Multifunctionality, we just try to summarize into a single metric, an index, the capacity of ecosystems to provide multiple functions simultaneously. And we did here, we use a very simple approach. We standardize different soil functions and productivity, and we, using the C-scores, and the average of all these functions was our index of multifunctionality, which in other studies we have shown that this index correlates quite well with other approaches. And here in this, and again, another structural equation model where we include both microbial diversity and multifunctionality, and of course, the links between them, and the links between these variables and other predictors, other important predictors like climate and soil pH of both microbes and ecosystem functioning multifunctionality. What we found is we found a strong positive link between microbial diversity and multifunctionality. And a very nice feature about these structural equation models is that when you find a significant link between two variables in the model, this link already takes into account the different correlations between the predictors. So it's somehow a measure of the effect of this variable in this other variable, in this case of microbial diversity and multifunctionality. But remember, this model is based on observational data. What we are doing is just fitting this model to our data. So that doesn't mean that the model is right, and it doesn't mean that there cannot be other models that are better, that can explain better our data. It's just a way to test particular hypotheses, which come with the relationship between the predictors with a statistical framework. And again, in this graph showing the importance, not a surprise, climate was particularly important. Increases in temperature reduce multifunctionality, increases in precipitation increase it. But again, it was particularly interesting to see the importance of microbial diversity, and in particular, the importance of fungal diversity to explain changes in multifunctionality across the global drylands that we survey. In later years, we have been quite interested and explored a lot nonlinear dynamics and nonlinear responses to changes in aridity. This is something that has been hypothesized quite a lot in drylands, particularly when dealing with issues like dryland degradation and desertification, and a bunch of models and theoretical studies have played with this idea of thresholds and alternative stable states, right? As you know, these theories suggest that when drylands become severely degraded or desertified, they change into another state, and once that you are in this degraded state, you cannot move back to the pre-degraded state because you have crossed a threshold. But these ideas have been mostly discussed from the theoretical point of view using mathematical models, but we don't have many empirical evidence of the presence of thresholds or alternative stable states in the real world. And we try to look for the presence of thresholds in ecosystem attributes in drylands, and we compile what in this study published on last year, we explore the presence of thresholds in over 30 ecosystem variables, including different microbial soil attributes of soil microbial communities. And here, we look at a bunch of things. We look at photosynthesis, nutrient contents in the soil and in the plants. We look at climatic attributes. We look at plant-plant interactions. Also, as I say, different attributes of microbial communities, like the relative abundance of mycorrhizae or pathogens. And what was really, really interesting about the results that we found is that all the variables that we look at and we use for doing this analysis, both data from microbial surveys and also data from satellites and other independent databases. So this was a quite thorough analysis. And what was really interesting is that we found the presence of thresholds, and not only one threshold, which is what has been typically hypothesized in the models, but we found 3 aridity thresholds. Some variables, mostly photosynthesis and plant productivity, change in a nonlinear way when an aridity threshold of around 0.5 was crossed. Then multiple soil variable change in a nonlinear way with an aridity threshold of 0.7 was crossed. And finally, other variables, particularly related to plant diversity and cover, and also to soil microbial pathogens living in the soil, change in a non-linear way with an aridity threshold of 0.8 was crossed. We can see here, for instance, to the right, some examples. For instance, at this aridity threshold of 4.7, we found a non-linear decline, for instance, in the relative abundance of mycorrhizal species. And for instance, an aridity level of 0.8, we found a non-linear increase in the relative abundance of fungal animal pathogens in the soil. We try to summarize all these changes in this picture. We try to illustrate how dryland ecosystems change from the, let's say, the less arid conditions. Remember, we are still in drylands, but in the less arid conditions of the global aridity gradient, we have enough drylands, enough rainfall, and drylands can function properly. They are productive. The plants do photosynthesis, incorporate nutrients into the soil. Soil microbial communities are diverse, are mostly dominated by beneficial species. And as we increase aridity conditions, we first cross this threshold of around 0.5, and then photosynthesis and the nutrient contents of the leaves decline in a nonlinear way. This is interesting because this nonlinear decline in photosynthesis happens without large changes, for instance, in attributes like cover or vegetation type. But once that this threshold is crossed, then photosynthesis is reduced, the organic inputs into the soils are reduced, and then things start to change. Vegetation starts to change, the nutrients in the soil, the soils start to become depleted in nutrients. So, and then another threshold around aridity values of 4.7 is crossed. And then the vegetation cover start to reduce. There is a shift in my soil microbial communities. There is also a shift in the type of vegetation. And as soon as this aridity threshold of 0.8 is crossed, vegetation collapse. Soil microbial communities become dominated by less beneficial species, more pathogens, and we have conditions similar to those as we have in the desert. So this is a nice example of illustrating how dryland ecosystem change in a nonlinear way to changes in aridity conditions, which of course this has implications to our understanding of how these systems may change under climate change. We also have used the soil samples collected from this survey to do additional experiments in the lab. This is also a very nice, wonderful resource for doing also all sorts of incubation experiments and other type of experiments in the lab. And one of the questions that we have addressed using these samples is thermal adaptation of soil respiration to changes in temperature. And this is a really hot topic these days in Earth system modeling and biogeochemistry, because the models currently being used by IPCC, they incorporate a positive feedback between soil microorganisms and temperature respiration. Increases in temperature imply increases in respiration, and this creates a positive feedback with climate change. The warmer the climate it gets, the more respiration, more CO2 is released to the atmosphere, so the higher the rate of warming. But there is a lot of debate about the magnitude of this effect and whether if this effect is global or not. It's very clear that it can be found in cold ecosystems, but it's not so clear that it can be found in other ecosystems. And typically, the way that we have approached this problem is either by doing short-term warming experiments in the field or by taking the samples in the lab from a given site and doing incubations at different temperatures. But our survey provides a really nice opportunity because we have across all the sites we survey, we have a 30-degree gradient in mean annual temperature. So what we did is we incubated soils from across this gradient in the lab at different temperatures and evaluated the responses without water limitations and without sugar limitations. We incubated them with plenty of water, with plenty of food, so microbes will work, will function without any limitation. And we look what happened. And what we found, this is a summary of quite complex analysis. What it shows here is potential respiration. And the results were quite interesting because, and this was not a surprise, if we increase the incubation temperature, respiration increased no matter what. It's not a surprise, we already know that. But what was really interesting is that respiration, no matter which incubation temperature we use, respiration was reduced as the mean annual temperature of the site increased. So, and this was observed regardless the microsite we use. We also have samples from under the canopy of vegetation, what we call vegetated microsites, and in areas devoid of perennial vegetation. We typically, of course, as you can imagine, areas under vegetation have more nutrients, are more productive. And again, hyperrespiration in vegetated areas, but the pattern with the mean annual temperature of the site was the same. So this suggests that soil microbial respiration adapts to ambient temperature in global dryland, so that the microbial communities of dryland ecosystems are adapted to the thermal regime they have experienced over the last hundreds, or at least a few thousand of years. And this is quite interesting because these results suggest that even with ongoing warming, soil respiration will not increase in drylands because first there will be not enough water, and second, soil microbial communities are adapted to the thermal climate that they currently experience. So these results are really quite interesting, and we further explore these patterns using models to try to understand the mechanisms underlying them. With our experiment, we couldn't, of course, we could guess which mechanisms were underlying the patterns that we observed, but we couldn't figure out which specific mechanisms were underlying them. So we use models, and this is also a very nice combination of field samples with laboratory incubations with modeling to try to disentangle the potential mechanisms underlying this case, the microbial respiration to responses to changes in temperature. We tested multiple models of different complexity. This is also again a very hot topic in this field, how to represent properly without being too complex the microbial dynamics into these models. And we found a very nice exercise, and in particular, we hypothesized that the carbon use efficiency, the relationship between carbon use efficiency, which is a key microbial parameter to define soil respiration, and mean annual temperature could be either positive or negative. So if it's positive, the model looks like this, and if it's negative, the model looks like that. Of course, if you only use models, you can get different set of results depending on your initial assumptions. But the nice thing is that we have empirical data to validate these model predictions, and our model or empirical data fitted really well a positive carbon use efficiency mean annual temperature relationship. So this suggests that that warmer climate selects for microbial communities with higher carbon use efficiency, as opposed to reduction in carbon use efficiency by warming. This is what many laboratory analyses laboratory experiments studies, short-term studies have suggested. And again, it's a nice example of combining field data, field samples with modeling and with laboratory incubation. We also have joined or dryland sampling, global sampling with samples from other ecosystems to make truly global surveys. And we also have been collaborating with multiple colleagues, different colleagues all over the world to conduct global analysis of the ecology and biogeography of soil microorganisms. The first study using this database, we have now the samples that I'm going to show here is with a database from 236 ecosystems from all over the world. And the first study was published 3 years ago. And it was very interesting because we observed that 2% of bacteria phenotypes accounts for over half of the total reads. So it's a measure of the relative abundance. So a very few OTUs account for most of the reads that you can find across global soils. We also related these abundant OTUs, these abundant soil bacteria, to different environmental variables to create the first set of global maps about microbial communities. And it was also a very nice study. So we identified particular clusters of bacteria that were linked to particular attributes, environmental attributes. Some were linked to high pH, some to low pH, some to high aridity conditions, etc. We did similar studies with soil fungi, and we found also very similar results. A very few fungi, less than 1% of the phenotypes we identify, account for over 20% of the reads. Again, a few fungal species are really dominant across global soils, and we explore some characteristics of these fungi that could make them so successful or at least so widespread. And again, this sets up with quite interesting results. We have been also working with protists in using these samples. And for instance, across global soils, we found that soil protist communities were dominated by consumers. And some interesting results is that their biogeographical distribution was largely regulated by climate. This was quite interesting because many studies have suggested that a key environmental parameters driving changes in microbial communities is soil pH. Soil pH has been, one of the most studied environmental variables, particularly in relation to bacteria. But in this global analysis of fungi, we observed that climate, particularly mean annual temperature and precipitation, rather than pH, was like the most important environmental predictor of the distribution of soil protist communities worldwide. We also have been exploring changes in specific groups of microbes. For instance, in this study, we look at created again these global maps of changes in plant fungal pathogens in the soil, and we found that they were strongly linked to temperature. Temperature was one of the most important environmental predictors of the distribution of these communities across the world. And because we observed this such a strong link with temperature, we also did some modeling to forecast future changes or potential future changes in these communities using a multi-scenario approach. And as you can see here, this is the actual distribution of plant fungal pathogens worldwide, and this is the forecast they forecast according to different models. And most of the models predict that because of increasing temperature, there will be increases in the relative abundance of plant pathogens in soil ecosystems across the world. So all of this is very nice. As you can see, it's really nice having these global surveys. They are really informative. They can provide important insights about the ecology and the biogeography of soil microorganisms. But one of the main questions and criticisms that we often get when trying to publish these results is that these are observational studies and it's difficult to get cause and effect relations. And of course, it's true. There is nothing really to say about it. It's just that with this, as you know, doing experiments at the global scale is quite complex and quite difficult, or in many cases, it's just impossible, particularly in many dryland areas. And we need to rely on these global surveys for gaining insights about the large-scale ecology and biogeography of these organisms. However, we also do experiments to try to gather experimental evidence or to see if there is experimental evidence for some of the patterns suggested by these global surveys. And we do a lot of experiments in my lab too, using plants, using microbes, and particularly using soil biograss communities. These organisms that I just love them, and we have been working with them for years, and are these communities dominated by lichens, mosses, cyanobacteria, liveworts, and other microorganisms that live on the top of the soil surface. And they are particularly abundant in dryland ecosystems. They can be found in many temperate ecosystems, but of course, they are particularly abundant in drylands because there are lots of open spaces not covered with vascular vegetation. We know that they play key ecological roles in drylands. They fix carbon and nitrogen. They regulate the hydrology of the system. They also regulate the germination and performance of many vascular plants. They increase stability, and they are the habitat of many soil organisms. So if we really want to understand the ecology of drylands, we really need to understand the ecology of these biograss, which are also very widespread. For instance, these are just 3 examples from Australia, from central Spain, or from Venezuela, where you can see like most of the biotic cover these dryland ecosystems is not given by vascular vegetation. Vascular vegetation in many of these places, they may cover 30%, 20%, 40%, 50%, but not many places you will find many more higher values of plant cover. And the rest of the soil, in the absence of disturbance, is occupied by these biograss communities, which, as I say, they are really important. So, for instance, we have used these communities as a model system to study the responses, ecosystem responses to climate change. We have several long-term warming experiments in Spain. And for instance, with some of the data we are gathering, we can test some of the predictions or some of the patterns that we have observed with these global surveys. For instance, in this graph, this comes from some of the results that we got from one of these sites, where we have been evaluating changes in soil respiration over time. So during the first 8 years of the experiment, we have been exploring in areas with a low degree of biograss development and in areas with high biograss cover, changes in respiration, in control, and warming environments. And we use open dome chambers, very similar to those used in the Arctic, to simulate warming. And for instance, we can see, but from the very beginning, particularly in the high biograss covered areas, how soil respiration is reduced. From the very beginning in areas with high biograss, and after the 3rd year in areas with low biograss. We collected soils, we incubated them and did another analysis in the lab, and we also gained evidence that the respiration per mass or respiration per microbial biomass is reduced with warming. So particularly in areas with low biograss cover. So overall, these results suggest that if you increase warming, respiration is reduced, and so microbial communities become adapted to changes somehow to the new warming conditions, and these patterns resemble quite well the global patterns observed, that if you remember is a decrease in temperature, sorry, a decrease in respiration with increases in soil temperature. We also have validated, for instance, the results of the global pathogen, the global survey of soil pathogens with experimental data, and we found that experimental warming after, in this case, I think we use again from another site, samples from 8 or 9 years of the experiment, I can't remember, or 7 years, I can't remember exactly the date, but again, we found increase in the relative abundance of all pathogens, of some major pathogens, and also increases in the total abundance with warming, which this was also a pattern that agreed with the observational pattern that increases in the relative abundance of pathogens with increasing temperature. And that's it. That's all I wanted to talk today. And of course, before finishing, I'm here alone, but this work will not have been possible without the work and collaboration of many colleagues. Of course, everyone in my lab, which is a wonderful group of young, energetic, and very clever, wonderful people. And of course, without all the international collaborators that are helping us and working with us for when doing all these global analysis. And of course, to all the funding agencies that have been funding our research over the years, and in particular the ERC, which has been funding continuously our research over the last 11 years. And thanks a lot for your listening. If you have any questions, I will be more than happy to try to answer them. Thanks.